Hello, my name is Zheng Zhang. I'm a MD PhD postdoc researcher working in Dr. Vijay Both's lab at the University of Pennsylvania. It's my pleasure to share my project at the third annual Immune Modulation and Engineering Symposium. My project is leveraging CAR T cells to achieve desensitization. As a brief background of my project, alloantibodies against MHC present a major barrier to transplantation, and the donor-specific alloantibodies preclude transplant due to the risk of hyperacute rejection. Highly allo-sensitized patients experience prolonged wait times and high mortality rate while awaiting a compatible organ. And the current desensitization approaches are ineffective as they do not adequately deplete allospecific B cells and plasma cells. Now CAR T cells therapy targeting CD19 and separately B cell maturation antigen BCMA have been approved by the FDA, and these two markers are nicely cover the B cells differentiate to plasma cells. So the goal of my project is to evaluate the efficacy of CAR19 and the CAR-BCMA as desensitization strategies. First, I optimized the CARs targeting mouse CD19 or mouse BCMA delivered to mouse T cells via retroviral vectors. As the slide shows, both CARs have a very high expression level. And then I tested the CARs in vitro by co-culture with antigen-engineered cell lines and primary B cells as well as plasma cells. As the figure 1a and b shows, both CARs show specific killing and a good amount of cytokine release. Then I treat the non-sensitized black 6 mouse with the CAR T cells. After 2 weeks of CAR T treatment, as the figure 2 shows, both the B cells and both in bone marrow and spleen, B cells was depleted after the CAR19 treatment, but they really remained a CD19 negative population of plasma cells. After the CD19 and the CAR-BCMA combo treatment, both the B cells and the CD19 negative plasma cells was fully depleted. And the figure 3 shows after the CAR treatment, the CD19 in positive cells in both spleen and bone marrow was deeply eradicated, as well as the CD138 positive plasma cells was shown after the BCMA and 19 combo treatment. Then I applied the CAR Ts in a allo-sensitized mouse model. What I do is I sensitize the black 6 mice with BALB/C skin, and after two times of sensitization, the donor-specific antibodies was tested by flow-based cross-match experiment. Then I treat the mice with CAR T cells or non-treated control. After 13 weeks, we can see in figure 4a, both non-CAR T treated group and the CAR19 owning group, their donor-specific antibody level is stable. And in the combo CAR T groups, their DSA dropped significantly, and at week 13, they reach a level close to a non-sensitized control. Then, to confirm the desensitization and test it in a more translational way, we made the same batch of mouse streptozotocin-induced diabetic model. After the STZ treatment, all the mice was successfully built diabetic as their blood glucose was over 300 after at least 5 days. And then the mice were treated with ATG to get rid of the T cells and let the mice have a T cell suppression. Then we then treat the mice with the biopsy eyelid. After the eyelid transplantation, we can see in the non-CAR T treated group and the CAR19 only group, their blood glucose reaches high, which means their eyelid was rejected hyperacutely. But in the combo CAR T groups, the mice blood glucose went to a normal level for a long time, and for each group we have more than 5 mL. And in the combo CAR T groups, we can see there are 2 mice reaches as long as over 60 days. So, as a summary of the project, pre-existing allo antibodies are a major barrier for successful organ transplantation, and we developed a preclinical syngeneric CAR T model to separately target CD19 and BCMA, efficiently depleting memory B cells and plasma cells. And the combination of CAR T therapy leads to effective desensitization, preventing hyperacute graft rejection. And B cells and plasma cell elimination pre-transplant may provide a window of opportunity to regenerate a more graft-tolerant humoral immune compartment after the transplantation to achieve long-term tolerance. That's all for my poster. And if you have any questions, I'm happy to discuss. Thank you so much.